Imagine an immense landscape beneath our feet, fungal genomes teeming with hidden treasures, small molecules waiting to be discovered. In this perspective-style exploration, we ask, how can we effectively pan for gold in mold, increasing the odds of finding novel bioactive compounds in fungal genomes? We draw directly from recent advances in fungal genome mining to outline strategies that prioritize the most promising biosynthetic gene clusters amid an ocean of genomic data. Fungi have gifted humanity with transformative small molecules, from penicillin to statins to immunosuppressants, yet many biosynthetic potentials remain unexplored. Sequencing of filamentous fungal genomes revealed unexpectedly large numbers of biosynthetic gene clusters, suggesting that perhaps only a small fraction of these gene clusters have been tapped so far. Yet most of these gene clusters remain silent under lab conditions, their products hidden unless we apply smart prioritization strategies. We group prioritization strategies under conceptual themes: leveraging known gene clusters relationships, hunting oddballs for novel scaffolds, following ecological cues, exploiting self-resistance markers, and looking to future computational and molecular tools. First, we consider strategies centered on known knowns and known unknowns, leveraging relationships to characterize gene clusters to find novel analogs. Many natural products exist as families of structurally related compounds, think statins or echinocandins, reflecting related gene clusters in different fungi. By querying databases like MeBig and using tools such as anti-smash known clusterblast, we can identify gene clusters partially related to known drug leads, prioritizing those likely to encode novel analogs with improved properties. Network analyses further refine this by grouping gene clusters into families based on conserved domains and highlighting those at intermediate distances for known clusters candidates for new analogs. For example, in the penicillin pangenome, conserved domains of PKSS, NRPSS reveal clusters related to known small molecules, but also reveal distant members worth exploring. Next, we hunt for oddballs, gene clusters encoding rare or novel structural scaffolds that might yield entirely new modes of action. Network analysis, again, helps by identifying gene clusters loosely related or entirely distant from known clusters. Alternatively, scanning for unusual domain architectures, PKSS with novel appended domains, or NRPSS lacking, canonical modules can flag gene clusters, producing unprecedented scaffolds. For instance, highly reducing PKSS with a C-terminal cat domain hint and novel release mechanisms, or NRPS-like proteins lacking condensation domains yield new alkaloid scaffolds. Likewise, unconventional terpene syntheses with novel cyclase domains can produce complex sesterterpenes, as seen in the discovery of quinolactone-type structures. Detecting unknown unknowns, gene clusters unrecognizable, Biomology requires tools combining genomic and transcriptomic patterns to predict novel clusters. Uncovering these could unlock entirely new families of natural products with unpredictable architectures and functions. Chemical ecology offers another beacon: gene clusters upregulated in ecological contexts, pathogen interactions. Competition, symbiosis often encode bioactive metabolites tuned to specific roles. Transcriptomic profiling during host infection can pinpoint silent but ecologically relevant gene clusters, such as phytotoxins upregulated in late-stage infection. Comparative genomics across pathogenic versus non-pathogenic species can flag conserved gene clusters likely involved in virulence or niche adaptation. Co-culture experiments, though challenging to scale, can reveal gene cluster activation via interspecies interactions. Transcriptomics then isolates the responsible clusters. Such ology-driven mining has uncovered metabolites mediating antagonism, e.g., antibacterial alkaloids repressed by bacterial lipopeptides in Aspergillus flavus. A powerful clue lies within the gene cluster itself. Genes encoding self-resistance mechanisms, often duplicates of the metabolites target. For example, the lovastatin gene cluster encodes a duplicated HMG-CoA reductase, and the mycophenolic acid cluster, a duplicated IMPDH, signaling the product's target. By scanning genomes for gene clusters containing duplicated core genes linked to known targets, we can prioritize clusters likely to encode molecules with specific modes of action. No single strategy suffices. Combining approaches network analysis, oddball detection, ecological triggers, resistance markers maximizes the chance of unearthing valuable small molecules. Advances in bioinformatics improve gene cluster prediction and structure function, prediction and molecular tools, efficient heterologous expression, and pathway activation will further accelerate discovery. Ultimately, the goal is predicting not only the chemical structure encoded by a gene cluster, but also its bioactivity, requiring expanded SMBGC databases and deeper biochemical understanding. As fungal genome sequencing grows through initiatives like large-scale fungal genome projects, our ocean of gene clusters will only swell, making effective prioritization ever more crucial. By panning smartly, leveraging known relationships, chasing oddballs, following ecological trails, and mining resistance clues, we can increase our odds of striking gold in mold. For researchers, integrate computational pipelines with lab validation workflows. Share curated BGCSM links in public repositories to enrich collective knowledge. For funders and institutions, support genome sequencing of underexplored fungal, taxa and platforms for sharing large-scale gene cluster network analyses. For viewers intrigued by fungal natural products, consider the vast potential lying in fungal genomes and how interdisciplinary efforts can transform medicine, agriculture, and beyond. Join us in the quest to pan for gold in mold, because the next life-changing molecule may lie hidden in a fungal genome, waiting to be discovered. Thanks for watching. If you enjoyed this deep dive, comment your thoughts on fungal genome mining, and share with colleagues who might be mining their own genomic gold. See you next time.